University Management. It's been a great honor for me, Novalia Kusumarini, to welcome you here. Before we go to the program, I would like to express our sincere gratitude to the Excellencies, Head of Soil Science Department of Brawijaya University, Dr. Syahrul Kurniawan, all of the lecturers of Soil Science Department, guest lecturer from University of Birmingham, United Kingdom, Professor Samiullah, and all of the audiences who follow this lecturing both from Zoom and YouTube. Ladies and gentlemen, this afternoon we will have a great opportunity to learn about response of forest soil to future climate change from the expert Professor Samiullah. Since climate change become a global issue that has a large impact to the human activities, including agricultural activities, many research have been conducted to mitigate climate change effect, and one of them are conducted in forests. Before starting the program, I would like to greet Professor Samiullah. Professor Samiullah, welcome to the University of Brawijaya in Malang, Indonesia. Even though you are here in UK today, but I'm pretty sure that your heart is here with us in Malang. It's a great honor for me to welcome you here. Thank you for your time, giving us a great knowledge and experience today. How are you, sir?
Doing good, and thank you for the invitations and introductions. Really glad to be here.
Yes, it's my pleasure. Hope everything doing great. And before we start the presentation, allow me to invite the Head of Soil Science Department, Dr. Syahrul Kurniawan, to give the welcoming speech. Dr. Syahrul, time is yours.
Okay, thank you very much to Novalia. Good afternoon, everyone, and also good morning, Professor Samiullah. I apologize if I cannot using the virtual background because at the moment the internet connection, the server in my house still restart, so I use my cell phone. So I apologize for that. Yeah, my name is Syahrul Kurniawan, and I'm the head of Soil Science Department, Faculty of Agriculture. On behalf of the Dean of Faculty of Agriculture, we would like to thanks very much to Professor Samiullah for have the time to give a lecture today to our student and also to the lecturer in the within the program of 2-in-1. So the 2-in-1 program is one of the flagship program of University of Brawijaya, and to improve the quality, both capacity and competence of the student, graduate, also undergraduate and also graduate student using learning resources from within Brawijaya, UP lecturers, and also the lecturers from abroad, namely all professionals who have an international reputation in their field. Also, the second aim of this program is to follow or to update the development in science and knowledge in their field through formal interaction with international professors. So by this program, we hope the material of the course today can enrich the learning material, especially for the course of soil fertility management. And also we hope it will increase the knowledge and competence of our students. I think that's all the introduction from me for the program. And thank you very much to all organizing committee who organized this meeting today. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Syahrul. Moving forward to the main agenda, we are really happy to learn about response of porous soil to future climate change. As I mentioned previously, climate change is a global issue that we should find a way to mitigate it. But in which part we should take part, or how forest soil can respond to climate change? Today, we will find the answers how forest soil responds to future climate change. But before starting the presentation, allow me to read the curriculum vitae of our guest lecturer. Professor Samiullah is Professor of Biogeochemistry from School of Geography, Art and Environmental Sciences, University of Birmingham. He gained the PhD from Louisiana State University, USA, and these are his teaching areas: environmental chemistry, analytical method and field research, physical geography, environmental management, and a lot of publications have been written, and those two are the most recent publication, which published in September 2021. Professor Ula, I'm sure that everyone is waiting for your presentation. We have time until 10:30 British time or 4:30 p.m. Indonesian time for listening to your presentation and discussion. For the discussion session, please raise your hand by clicking raise hand icon in Zoom room, or type your question in chat room. Then I will mention your name and you can start asking your question. For the audiences who follow this lecturing in YouTube, you can type your question in comment room and I will read to get answers from our guest lecturer. During the presentation, please stay mute your microphone. Now it's time for listening to the presentation. Professor Samiullah, time is yours.
Thank you very much, Nevelia, and thanks to the Department of Soil Fertility and Soil Sciences. And I'm really glad to be here and share some of the preliminary results of our experiment in terms of the response of forest soils to climate change. And as you mentioned, would be happy to go and answer questions after my presentations in relation to the work that I'm presenting on behalf of the Institute. So let me first start sharing my screen. Noelia, can you hear me clearly and can you see the screen?
Yes, we can hear and see it clearly.
Okay, then. Thank you very much for the invitations, and really happy to share some of the soil science research and forestry research that we do at Birmingham University under the Birmingham Institute of Forest Research this morning with you all. The focus of my presentations would be a 3-year carbon dioxide enrichment facility, which we call a FACE facility. This facility is evaluating the response of temperate forest to elevated atmospheric CO2 concentrations. And this work involves hundreds of researchers, so I'm presenting their work on behalf of the BIFAR overall. Before I go into the science, I want to introduce the University of Birmingham, which is located in the middle of England, about 120 miles north of London. And it's a young city in Europe and have a lot of green spaces around. So you could see in the background picture that Birmingham city is really green. It is one of the greenest city in Europe in terms of the number of trees. So quite a fascinating place to be in. Now the University of Birmingham, it was founded in 1900 and it's a leading UK red brick university. It's a comprehensive one, so it's with intensive research in international reputations. Birmingham University is a member of the Russell Group, which is leading research intensive universities in the UK and in the top 100. And the university have alumni and former staff that includes about 11 Nobel laureates. The main campus of the university is in Birmingham, and recently we have another campus opened in Dubai. This is a very diverse university. 34% of the academic staff are from international destinations. And then our student population is between 30,000 to 35,000. And usually we have 7,000 to 8,000 international students, undergraduate, master's and PhD students. The university have 5 colleges, and one of the colleges that my school belongs to is the College of Life and Environmental Sciences. And then I'm based in the School of Geography, Earth and Environmental Science. We have the School of Biosciences, and between the School of Biosciences and Geography, Earth and Environmental Science, we have the Birmingham Institute of Forest Research that I represent. The School of Geography, Earth and Environmental Science have 95 academic staff, 1,200 undergraduate, master's and PhD students. And again, the original department was established in 1900 as a Department of Earth Sciences, and then it's expanded to Geography and Environmental Sciences. It has quite a diverse expertise in key research areas, particularly environmental change and biogeochemistry. We have a stream in nanotechnology and precision agriculture, including work on developing nanofertilizers, a strong group here in atmospheric chemistry, in the air quality, and water sciences in a changing world is another stream that is very strong in here, in environmental health sciences. And the 6th component is the Birmingham Institute of Forest Research, which cut across the various colleges and departments within the university. And the institute in its experimental picture, you could see it on the right. So the Birmingham Institute of Forest Research is a broad interdisciplinary initiative of the university to provide fundamental science, social science, and cultural research, which is of direct relevance to the landscapes, particularly forested landscapes anywhere in the world, to really make it sustainable and deliver the functions, the multiple functions that the forested landscapes deliver in terms of ecological functions, societal functions, and even cultural. Part of this institute by far is we have the pre-air carbon dioxide enrichment facility, the FACE facility, to evaluate the response of forests to future elevated atmospheric CO2 concentrations. Within the institute, we have the Forest Edge Doctoral Scholarship Program, which is PhD studentships with multidisciplinary focus on unforested landscapes. So we have students in the English department looking at forests and its cultural aspects. And then we have students looking at the physics of forest and the dynamic of wind flows around forested landscapes. So it's a very, very multidisciplinary forestry program. In terms of research, it goes from plant biology genes to the landscape perspectives, and biogeochemistry from typical nitrogen, carbon, and phosphorus to micronutrients and macronutrients in its cycling under climate change. So quite a diverse research portfolio the institute have. With the introductions gone, I now want to switch mainly to the questions of soil carbon stocks and climate change and rising atmospheric CO2 concentrations. Globally, in soils, which is a critical source for the storage of carbon, the soil organic carbon, and in the upper 1 meter, we have about 1,846 petagrams of carbon. And if we consider 2 meter deep soils, then we got about 3,012 petagrams of carbon stored in these systems. We also have the soil at inorganic carbon stocks up to a meter depth, which is 940 petagrams globally. And if we think of the soil carbon pools in 1 meter, 2 meters, in even the inorganic soil carbon pools relative to the atmospheric CO2 pool, then we do see it's the atmospheric CO2 concentrations in pool is a fraction of the soil carbon stocks. So soils are very critical for storing and sequestering the carbon. It has been doing that functions. The challenge that now we have that most of the time, originally we had natural ecosystems populated by forests and peatlands and subsistence agriculture, so where we had a larger capacity for soil carbon storage, but with the rise in populations and the demand for food in agricultural productions, mostly in the world, we have drained, ditched, and cleared natural ecosystems to make it cultivable for food in cash crops. Doing so, it enabled and ensured food security, but at the same time, we lost carbon from the soils. And this figure shows thousands of years of the history of the used land for agriculture or agricultural land uses under grazing land in green color, cropland in the other color, and the yellowish line color is showing the soil carbon losses. Time on the x-axis and on the y-axis, you could see land area under cultivations and how we changed. And then on the y-axis on the right, you could see the cumulative soil organic carbon losses in petagrams of carbon. And these cumulative losses are over the 2 meter depth of the soil. So overall, we have lost 133 petagrams of carbon since human dominated the landscapes and gradually converted natural ecosystems to cultivable lands. You will see that grazing land seems to be contributing more into the land area, but and more to the carbon losses, but that's because it's more extensive. So the carbon loss from grazing system is relatively lower than the cropland systems, but the land area under grazing land is bigger. So that's why the contributions in terms of SOC losses looks bigger. But most of the carbon in the intense losses are mainly from arable agricultural lands or croplands where we do continuous cultivations. So 133 petagrams of carbon we lost into the year from the soil pools. I'm not covering any inorganic carbon losses in the threats to those carbons, but if there were interest in questions later in the discussions, I would be happy to talk about that as well. If we consider only 1 meter of the upper soil carbon, the upper soil carbon loss stocks, then it's about 75 petagrams of carbon that we lost globally from these terrestrial landscapes. So there is opportunity to really manage the land in a sustainable way, diversify our landscapes, and enable the soils again to capture the carbon that we have lost. Of course, there is a limit how much it could store, but there is still the potentials that we could ensure to enhance the capture and sequestrations of carbon in landscape, including cropland, grazing land, forest, peat, pastures, all these systems through alternative soil husbandry and soil management techniques to really ameliorate climate change. Now I switch on to just the anthropogenic fluxes of the carbon dioxide, why carbon sequestration is important. The key is that we have fossil fuel emissions, that's 9.3 petagrams of carbon per year. In the red arrow, you could see it. Then we have the atmospheric growth of the CO2 is 4.5 petagrams or gigatons of carbons per year. Land use change, whether it's fire, conversions of one land use type to another is about 1 petagram going up. And we have about 3 petagrams or gigatons of carbons land sink into the terrestrial systems through the capture of carbon dioxide by vegetations. And this is bigger than the oceanic sink. So if we look at the budget, we are increasing the atmospheric pool by 4.5 gigatons, and 3.1 gigatons are taken up by the land through the process of photosynthesis, mainly. So again, this land sink have the capacity to be maintained and even enhanced to really arrest the atmospheric growth and lower it down if we have to stabilize the atmospheric concentration of CO2 through the contributions of the land systems. Of course, there have to be control measures and reductions in the emissions of CO2 from industry and fossils and those sources, but the land sink is not insignificant. And more importantly, the land sink of carbon is important not only to capture the carbon, but the organic matter in soil determines the health of the soils itself. So healthy soils relies on good amount of organic matter in organic carbon to allow the biodiversity, the soil biodiversity to thrive, to sustain the fertility of the soil systems, to really support higher infiltration, reduce flooding in the landscapes, and to produce food sustainably. So these are the key benefits of having more organic matter in the soil systems. Now, this land sink, if we look at the total emissions into the atmosphere in a simple picture here, the total anthropogenic source emissions that goes up into the atmosphere, 1/3, as I mentioned in the previous slide, about 30 to 33% of that emitted CO2 is taken up by the land ecosystems, the crop systems, the wetlands, the peat, in the forest, and everything. So it's 1/3 of those emissions. So it's really an important carbon sink. 26% again by the oceans, and there is a 44% increase in the atmosphere. Now, the land carbon sink is more potent and sequestering more carbon in the northern hemisphere. In the forest in the northern hemisphere absorbs or sequesters 3 billion tons of carbon dioxide per year. And within the northern temperate, within the northern forest, which is boreal and subarctic and temperate forests, out of all those types, temperate forest absorbs about 0.8 billion tons of CO2 per year. So that's a substantial amount. The northern temperate forest carbon sequestration is pretty much equivalent to the total CO2 emissions from anthropogenic sources from the EU 27 nations. So that's a substantial sink. If not, then the amount of carbon sequestered by northern temperate forests is equal to the total amount of CO2 emitted from global deforestation. So it's a substantial carbon sink in the northern forest, among other land use types like tropical forest and boreal forest. But here I'm tailoring more and focusing now on temperate forests. The question now, is as atmospheric CO2 concentration increases, you will remember or recall that pre-industrial CO2 concentrations were 280 parts per million. Now we are touching around 14 parts per million in our atmosphere. So because carbon dioxide fertilizes our plant systems to capture it through the process of photosynthesis and sequester it into biomass and soil carbon. So the atmospheric CO2 concentration have increased, and it is likely to increase. So there have been modeling studies looking at how the different climatic zones and the different crop or forest types might be responding to increasing CO2 concentrations. In the figure here, I only point the deciduous broadleaf forest, DBF here. The response, it's beta sensitivity. It's the sensitivity of the productivity to changes or increases in CO2 concentrations. So that's that sensitivity is quite high, and it does, when the CO2 concentration increases, this study show that CO2 uptake by deciduous broadleaf forest would increase as well. And you, on the right side in the figure, however, how long this uptake of CO2 under increasing atmospheric CO2 concentrations will persist. On the right side, it shows that it might decline. So there is this dotted dash line that over time it might decline because there is a capacity that forest may no longer be able to sequester that carbon over long time scale. So of course there is this time sensitivity and time factor into this. But overall, these forests are responding very well into enhanced carbon capture when they're exposed to elevated atmospheric CO2 concentrations. These studies, modeling studies, points to this long-term carbon capture increase by forests. And so far, we don't have much experimental evidence. And the reason is that it's very difficult to have a phase experiment, a 3-year carbon dioxide experiment, to really undertake the type of research in elevate carbon dioxide to look at the response of forests. And the other thing is, even if you elevate carbon dioxide, the response in a year, 2 years, 3 years might not be sufficient to enlist the long-term changes in the carbon balance of these ecosystems. Previously, there were phase experiments which looked into the response of those forests, but mostly those phase experiments will work on young or plantation forest planted on formerly agricultural land. But none of those experiments considered mature temperate forests, mature non-fertilized systems, to see really if carbon enrichment in the atmosphere would really enhance CO2 uptake over time, or whether there could be other growth limitations that might limit the CO2 uptake potentials to really tweak and come up with the more robust and less uncertain estimates of carbon budgets globally under future climates. So as I mentioned, the previous phase experiments in Paris, like here is a picture of the Oak Ridge sweet gum liquidemba phase experiments in Tennessee in the United States of America. But it was done on young plantations rather than mature one. And the plantation was undertaken on a non-forest soil. Young forest grows very vigorously, so the responses are CO2 uptake projection based on those young forests. Seems to be not directly applicable to mature forests, which are more than 50 years old, because the responses to CO2 increases could be different. There would be different nutrient limitation scenarios and priorities, and how further growth might be limited by nutrients, where nutrient cycling in mature forests without fertilization seems to be very tightly coupled. So with these questions in mind, how mature forests would respond to increasing atmospheric CO2 concentrations, particularly the concentrations of atmospheric CO2 expected by 2050. So bifurcates is that preemptive place where we recreate the future atmospheres around forest patches, and then the intent is to see how the forest would respond to these fumigations in terms of the CO2 fertilizations, and what if there are further growth limitations by nutrients, water availability, or even pathogens and diseases. So all these aspects are the focus of this experiment. Our 3-year carbon enrichment facility have 3 non-infrastructure control plants where there is no pylons or pipes that you see in here, if you could see my mouse. And then we have 3 ambient phase rings where we have all the infrastructure and we just blow air through it, but no elevations of carbon dioxide. And then we have 3 treatment phase rings, which where we elevate or increase the concentration of carbon dioxide by 150 ppm above the ambient. So if the ambient concentrations in the morning is 400, then the treatment rings would have 550 ppm of CO2 from the ground vegetations all there to the top of the canopy. And we started these experiments in 2015-16, where we generated baseline data. And then from 2017, the fumigation started. Our current plan is to be doing this fumigation experiment until 2026, 2027 to have a decade of fumigations. But the by far ambition is to go beyond a decade, because mature systems may even take longer to adjust to this new reality and to really be able to enlist or detect changes, because ecological changes could be very subtle. And so that's why we need a longer experiment. And so our ambition is to really go beyond a decade and maybe at least run these experiments until 2030 and beyond, depending on how much funding we could secure to keep doing this experiment. Is a pictorial of the plots. So the one with these circles around it is our elevated CO2 fumigation plants, and then we have the other three, which are the control one. And the work published, so we pretty much delivered the higher concentration. You could see it in the figure in the top corner, the red colors, where we pretty much delivered 550 ppm CO2 into the treatment rings. And 95% of the times we were successful in doing that. So it's published, and our experimental setup and the way we fumigated it, it's quite robust. The work is published if you want to read further by Hart et al. And the residence time of the CO2 that we fumigate into these forests is about 3 minutes, but it's all fully automated. There are sensors for carbon dioxide across the length of the canopy, and then based on those concentrations, the fumigations and directions of fumigations, depending on the wind direction, is adjusted. So we have many questions, primicology, biology, physics, and everything that you could imagine in terms of forest ecosystems and their management to address under this experiment. Today, I will only focus on some of the key questions that is related to the trees itself and the below ground processes, nutrient cycling. And the reason I pick up nutrient cycling, I'm a soil scientist myself. This is where I do the bulk of my work. And I'm speaking to you all in the soil science and soil fertility department. So that's more relevant here. And the key questions we asking, whether trees under elevated CO2 in mature temperate forest would be able to allocate more carbon resources below ground, and whether those allocations would allow the soil nutrient cycling processes to furnish or provide more nutrients to the trees to really capitalize on the elevated CO2 and enhance photosynthesis. So unless we have nutrient surplus, there might be limitations of the CO2 uptake potentials. So these are some of the key questions, and we are also looking at the overall budgets of nitrogen, carbon, and phosphorus at the ecosystem scales. This is a young experiment, so only the fumigation started in 2017, so we haven't published much of the work, and a lot of colleagues are involved, but today I will share some of the key findings from this experiment that have come so far. The first key one is the above ground tree response to elevated CO2 fumigation, or the ECO2. In the figure, we show the flux density of the photons at the light intensity, and then the A net is the uptake of the CO2 in micromoles per meter square per second. So we looked at that. And because it goes from a low intensity in the lower canopy to increasing light intensity in the top canopy, overall in this forest systems, which here is an oak-dominated forest. And the oaks are 150 to 170 years old. So these are really old, mature, old trees. Overall, this forest over the last 3 years sustained an elevated CO2 uptake. The maximum effect that we detected so far is 33%. So 33% more CO2 is taken up by the ECO2 fumigated plants compared to the control. You could see it in those figures. And the paper is published and available. If you want to have a copy, I could send that as well. The overall average CO2 uptake over all light conditions from sub-canopy to the top canopy is about 23% more CO2 was taken since 2017. So that's an evidence that as you increase and as we reach the atmospheres of 2050, these mature systems would be able to take up more CO2. But this finding is only based on 3 years of measurements, and we will be looking more and more to see how far the forest would be able to really sustain this higher uptake, to be able to more accurately model and predict the land carbon sinks, particularly in temperate forests. And we are collaborating with similar experiments in Australia, which are looking at eucalyptus plantations, eucalyptus forest under ECO2, similar setup. So at least we could come up with some global approximations. There is an Amazonian forest phase experiment. The infrastructure is there, and they are working towards starting the Amazonian phase. And they have some experiments already going on, and we are actually collaborating. So at least we could cover the 3 major forest types globally. But in this case, temperate forests, we do see an enhancement of CO2 uptake by the trees. This is very preliminary unpublished data, but we doing breast height diameter measurements and tree measurements. Every tree in this forest is tagged, and it comes with dendrometers, automated and manual. And this is very preliminary, but there seems to be some evidence of more NPP by the elevated CO2, which is in blue color here, compared to the ACO2, which is our control plots up to 2020. So more carbon uptake, and there seems to be relatively more NPP in this mature system. Now from the above ground, I go to below ground. So the one thing we looked at the soil respirations using automated glycol systems. It's also coupled to a Picaro greenhouse gas analyzer. So we're looking at the fluxes of methane, nitrous oxide, and we are continuously measuring CO2 exchanges. So we're doing soil respirations, and we are also attempting to partition heterotrophic and autotrophic soil respirations as well. This is ongoing, but the pre-treatment year 1, then the dormant in the second year, soil respirations in micromoles of carbon dioxide per meter square per year. It's unpublished data by Angeliki Kormuli, who just finished her PhD and working towards submitting this paper. But in the initial years overall, we detected 20% increased soil respirations. And we are further exploring whether the soil respiration increased mainly by root respirations or fungal respirations or bacterial. So we are further partitioning some of those soil respirations. At least we will be able to partition autotrophic and heterotrophic respirations to see whether there is enhanced root respirations or whether it's both roots and the microbes in the soils that are respiring more. So there is more CO2 uptake, maximum 33% in the upper canopy by oak forest. But remember, we have some other vegetations we haven't been able to measure it yet. And there is some herbaceous vegetations as well. But at least from the dominant oak tree perspective, more CO2 uptake. In the initial years, we see an enhancement of the soil respirations. In the later years, the data is not finalized, but we're seeing calming down of this difference in soil respiration. So the control in the treatment plant seems to be converging to less of a difference than the 20% that we detected initially. So more soil respiration, at least at this stage, means higher microbial activity in the soils, and this might be indicative that the trees are allocating carbon below ground really to enhance microbial activity and seek the nutrients that are ultimately critical for carbon uptake. Kuliah, is it? Can you guys hear me clearly? And it's going fine. Okay, I didn't hear a response. So here I'm showing the roots. What's happening to roots, which are less than 1 millimeter in diameter in the soil horizons in these forest systems. So these are fine roots. So we have been measuring the fine root dynamics through mini rhizotrans, and then we also had the standing stock and stuff, but this is from the mini rhizotran. In the O horizons in the top panel, A horizon in the middle, and then B horizon at the back. And we started doing that from 2018 all the way to 2021. And then the blue color is the control plots. The orange is the elevated CO2. And the green is the undisturbed plots, the plots that doesn't have the infrastructure. In the O horizon, we see a substantial increase in fine root biomass, which are less than 1 millimeter in the O horizon. This is not significant in case of the A and B horizons. So it seems to be pretty much similar. But in these forests, the bulk of these fine roots are in the O horizons, in the top organic layer. There is more probably the potentials for later in root decompositions in nutrients availability. So it might be the first initial response that the trees are now having more fine root biomass to really secure the nutrients. So that's one evidence of that elevated CO2 fumigation impacts going below ground. So we saw enhancement of soil respirations, and we see higher root biomass. So if there is higher root biomass in the O horizon, of course, the associated respirations might be the reasons for some of the enhancement of soil respirations overall that we saw in the previous slide. We currently, so this is the project I'm leading on root exudations and looking at the metabolomics of roots. So it's very fresh data, but we installed the root boxes into the forest floors with windows that you could see window here and window here. And then if we remove these insulating materials, then you could see the window and how the roots and the fungi and everything that grows. And we could see some of the mycorrhizae in these light white colors that are associated with oak roots. So we installed these root boxes on site to really track particular roots, collect the exudates, even though it's a disturbance, but at least from intake roads, and then look at the concentration of carbon, nitrogen, and phosphorus in the exudation rates, quantify the exudation rates, and also look into the metabolomics to see if the chemistry and the concentrations of carbon, nitrogen, and phosphorus exudation might change under elevated CO2, targeting nutrient acquisitions. So that's the eventual aim, whether root exudation might be priming the microbes in the rhizosphere to really go and mine further nutrients in the soil environment, being a mature system. So we started root exudation collections over time from this in 2020, in the middle of the COVID, but we were able to access our site and we had the first exudation collections, and then we continued with that. The approach was initially when we started was to identify the oak roots. Of course, there are other roots, and then wash it and then acclimatize it for 24 hours in glass beads in nutrient solutions. And then after that, 24-hour incubation period, we then changed the nutrient solutions and collected the exudates that way, transported to the lab for further analysis. The root that we used for exudations, we also clipped that root, and we did the morphological analysis of those roots through the Rhizovision Explorer, so that we to be able to normalize the exudations of carbon and nitrogen by length or surface area or mass. together. So this is the root exudations of both dissolved organic carbon and dissolved nitrogen. And that's started in August 2020. So August, November, March, and June of this year. March and June of this year, this is what we collected. And then in the blue color, we have the control plots, and in the orange, we have the elevated CO2. So the exudations of carbon in general, and particularly in August and March, was significantly higher, reaching up to 37% more carbon exudations from the treatment plants compared to the control. In November, it wasn't significantly different for the DOC, but at least there is a tendency towards more exudations from the elevated CO2 plots. We did not detect any substantial changes in DOC content. Even the rates were lower in June this year. And we still undertaking these studies to look at the overall dynamics of these exudations and then related to the standing biomass of 5 roots to really quantify the total rates of carbon and nitrogen exudations. What we see in DOC, that the dissolved organic carbon, we see at least changes in the rate of exudations. This change was not very stark or significant in case of the total dissolved nitrogen, so it pretty much remained variable and similar across the treatments and across the monthly, the seasonal sampling campaigns. Here we show the carbon to nitrogen ratios of the exudation. So of course, if the DOC increases in the nitrogen does not increases in terms of exudations, the DOC to TDN ratios is ultimately then changed, and we saw a more substantial change in the August of 2020. Overall, there is tendency for higher DOC to TDN in the elevated CO2, but only significant in case of the month of August. The C2N ratios of the roots that we collected the exudates from is also shown on the right side in the plot, and it did not change so far. So there is some evidence of more carbon exudations, maybe less of the nitrogen. That might be one strategy, that the trees are now trying to conserve nitrogen and maybe exude more carbon below ground to prime up microbial activities. And for this, we sent up and analyzed the exudates that we collected in August 2020 for metabolomic analysis. We only at this stage did untargeted analysis of root exudates in roots. And the data is very preliminary. It's not finalized yet, but I just wanted to share the glimpse of what's going on. So the negative ions or signals of these untargeted metabolomics really detected stark differences between the elevated and the control plots metabolomic signatures. This is a huge data set, so we are still doing a lot of the analysis to really come up what have changed and what are the key driving compounds or metabolomes that have changed substantially. And so those analyses are underway, but the initial data suggests that the chemistry of the exudations change in response to elevated CO2 for the negative ions from the mass spectrometry, for the positive ions, it's similarly. So there are these stark differences between the two systems. But this is an ongoing work, and it's very, very preliminary, but we see a change in exudation rates, and we see a change in the chemistry. Finally, we are focusing to quantify and tweak the change in chemistry so we could produce an artificial cocktail of exudates and then do a further experiment to see how it might be mobilizing nutrients. At the same time, we looked at later decompositions under elevated CO2. So we had sieved homogeneous soils in each experimental area. And then we had root and mycorrhizal exclusion cores. So 2 millimeter would allow the roots, 41 millimeter would exclude the roots, but allow the mycorrhizae to go in to the soils, and 1 micron would exclude both roots and mycorrhizae. And within each core, then we had either root or leaf litter from the same forest type. We buried it here. You could see it in the picture. And we started these time steps collections. So it's an ongoing work that we, from time to time, at least after 3 months, we collect these cores, the 2 litter types and 3 mesh types, and then we look at the amount of % change in the dry matter of the material that we buried to quantify the rate of decompositions, and we also look into the enzyme functions and nutrient availability. The figure here shows the dry mass, and then on the x-axis we have the 1 micron, 41 microns in 2 millimeter sizes, and then we have leaf and root material. It's highly variable, and it's just 3 and 6 months after the burial, so it might take longer. It's a long-term experiment, but we buried these in October, and then we collected it in March. The only thing we could see currently, it looks like the roots seems to be maybe it seems to be decomposing faster than the leaves, but at the moment it's not very significantly different to claim that, but there seems to be a tendency. Too early to call it, but this is my personal view that maybe the roots are the dynamic pool that would decompose much faster than the leaves, the way we saw the data is coming through. Here is, we also looked at the extracellular enzyme activity in the soils in these exclusion cores, and the x-axis you could see it in 1 micron, 41 and 2 millimeter cores. And then for March in 2021, in August 2021. And then we looked at the beta glucosidase on the left side. We did not see any key difference, except there was a significant difference in the beta glucosidase in the cores that were for the root material in the 2 millimeter, where roots are allowed to go. So maybe the roots enabled some higher activity in either the exudations, enhance the beta glucosidase, but this work is continuing and we will have to see. But overall, the picture is that we did not enlist a key difference in the beta glucosidase enzyme activity. The acid phosphatase, on the other hand, to the right side, activity for the 41 micron in the root, and then for the 1 micron in the root, they had opposing effects. So there are some changes, but overall the patterns are similar. So we don't see much difference in these extracellular enzyme activities in the very beginning of this long-term decomposition study. So it might change, but this is so fresh, and we have yet to see the long-term dynamics of these enzyme activities to see whether that might change to support nutrient acquisitions. We looked at potential grass nitrogen transformation rates using 15 in nitrogen tracing techniques, collected the soils from these cores in 2018, 2019, and 2020, brought the soils into the lab, homogenized it, and did lab incubations. Now, from 2021 onwards, we started this experiment in the field, so doing incubations in intact soil cores in the field. But here we see the gross mineralization rates in this slide to the left. So the treatment versus the elevated CO2. In general, there is a tendency for mean higher gross mineralization rates in the elevated CO2 plots. Non-significant except in 2019, where the rates were lower, but significantly different between the control and the treatment plots. But there is this tendency of relatively more mineralization rates in these plots. If we look at gross nitrification rates, it remained non-significant except in 2019 as well. The microbial immobilizations is also pretty much similar, except in 2019 that we saw a significant difference in the immobilizations of nitrate and ammonium by the microbes in the 2019 year. This is interesting, and I don't have time to go into details, but the year-to-year variations in how wet or dry the soil was. In 2018, we had some insect infestations of the leaves, which were just debarking the leaves, and there were more DOC being released. So really, really important here to note that a year or 2 or 3 year study sometimes may not really reflect the long-term picture of these changes, because this variability we see over the 3 years is not giving us a very clear clue of where we are going. We looked at the potential into emissions to see if the trees would need more nitrogen. Nitrification might enhance, or nitrogen cycling might enhance, and such cycling might promote the emissions of N2 and N2O productions, the nitrous oxide gas. And then we wanted to see if what would be the relative contributions of the denitrifications to the total N2O productions in these systems. This is the one thing that each time, originally, I personally hypothesized that N2O emissions might be reduced from this forest because the forest would be looking for the nitrogen, and somehow the overall plant microbe interactions would really make it tight so that not much nitrogen might be available to lose it to groundwater, and maybe to lose it to the air in the form of N2O, NO, or dinitrogen gas. But based on the lab studies, we always see on average more N2O coming out of the elevated CO2 system, showing some upregulations of the nitrogen cycling processes. And on the left here is the denitrification source. In 2019, it was the overall rates were low, but there was more from the treatment plants than in the control one. If we look at the nitrification source into the nitrification source into presented similar patterns to what the denitrification source were, but overall denitrification seemed to be the more potent and dominant pathway of N2O productions in these forest soils. And we did some further studies. Don't have the time to show that data, which was a different setup, not the isotopic one, where we also saw a relatively more N2O emissions from the treatment plant. So it does indicate a change in the soil nitrogen cycling processes, at least in response to elevated CO2 fumigation. And if the soil microbial activities are changing, so we also over time, seasonally, 4 times a year, we measure the microbial biomass, carbon, nitrogen, and phosphorus. Here I'm just showing the microbial biomass, carbon, in the top panel, which is the O horizons, and then the middle is the A horizons, and the lower is the B horizons, and that's from 2018 to 2021. I don't have all the data in hand, but only we see a substantial increase in the microbial biomass, carbon in the O horizon, where we have the bulk of our fine root density. And if you recall, we also saw an enhancement of the fine root biomass in the O horizon. We also see an increase in microbial carbon in those horizons. So it does indicate higher microbial activity, potentially higher carbon allocations below ground. And then that's probably the results to really, the roots probably is in search of the nutrients, because we believe these forests have to be co-limited by both nitrogen and phosphorus. So this is a ongoing experiment. There is a lot going on. I only was able to cover some of the biochemical aspects of this experiment. And the ultimate aim would be to really put this tree in the middle and do the overall budget of nitrogen, carbon, phosphorus, as well as water and the associated processes. This is a work in progress, but so far, what we see is an enhancement of the CO2 uptake in the upper canopy of the oak, an increase in NPP above ground up to 15%. We see some increase in litter. I didn't show the data yet, but the litter part, but this is an ongoing work. And once we have some confidence in its presentation, then in its final, it will come through, but we do have some evidence of that. We see a 45% increase in fine root NPP, 50% increase in microbial biomass, carbon, particularly in the O horizons. We did not detect much changes in the water. Originally, we thought that stomatal conductance would be reduced, and we will have probably more moistures in the elevated CO2 flats, but we haven't detected that change yet. I did not show data, but we saw the enhancement of biological nitrogen fixations by the non-symbiotic co-horizon layer. And overall, we have detected a 20% increase in the soil respirations in the initial years of fumigations, enhancement of root exudations, in changes in the exudate chemistry, and we see some changes in nitrogen mineralization and immobilizations, and some enhancement of into emissions. So that's pretty much the picture that I have for now, but hopefully in future, I might be able to come back and fully substantiate some of these changes and maybe bring in some more confidence in terms of long-term studies. So confidence means when we have more data and we are able to really pinpoint the potential changes and then be able to do the budget. Our ultimate goal is to do the ecosystem scale budget for carbon, nitrogen, phosphorus, and water. And that information would help modeling the response of us to elevated atmospheric CO2 concentrations and how much carbon it could take and whether there would be limitations. We have now substantial funding received to look at the microbial aspects and tree diseases and pathogens. So that work will come along, and then hopefully we'll be able to see if under future atmospheres or climates there might be more risk of tree diseases, and that might jeopardize the potential that we see now in carbon uptake. So that's it's a huge work done by the Department of Biosciences in here, and quite an interesting stuff going on there, including looking at if the tree have some immunity system and they have the memory for the immunities to really adapt to climate changes if there are new pathogens or new disease risks. This work that I presented on behalf of the BioFAR involves a lot of global collaborators, so I cannot go through individuals, but you could see that we have substantial national and international collaborations. And this is the goal of BioFAR, that it's not about the phase experiments. We're doing research in Amazonia and elsewhere in the world in forest, and like coming to Kuliah University, Indonesia, is with the same motto that we want to collaborate in research in forested landscapes. The work we presented involves a lot of the researchers, so I can't go through the names of everyone, but everyone is acknowledged here. We have some of the results presented in details in our Treescape conference in 2021. If you like, and you could find that. Some of our latest results could be found through the links, and I could sing this BioFAR communication links to you guys if you are interested to follow up. We have a conference, Trees for the Future, to look at the diversity and complexity for resilience in carbon storage. That's next week almost. So it's on campus, but it's also virtual in case there is some interest from your end in participating in this conference, which looking at the trees and forested landscapes from science to policy to management and future directions. I think with this, I will stop and thank you all for your patience, because it's a long talk, 60 minutes or so. But I managed it in time. So thank you all, and I'm happy to take up any questions.
Thank you very much, Professor Samiullah. I'm sorry for not responding you earlier because it's stormy here. So I got several issues related to signal and your voice that cannot clearly catch by me previously. But thank you very much for the fruitful presentation. You mentioned that Birmingham University has forest facilities named B4, and Brawijaya University has also a forest facility named UB Forest, and we're lucky the chief of UB Forest, Dr. Cahyo Prayogo, is joining with us, and perhaps we can have a warm discussion later on after the seminar. And also you mentioned in chat room that you see your colleague from UK, and yes, we have a collaborative research with CEH start from 2 or 3 years ago and still running. Thank you very much, Professor
Thank you.
Samiullah.
Just to make a point that I showed the forestry research and forest biogeochemistry, but agriculture, because your department is about fertility in agriculture, so we have a strong component in agriculture soils and the biosciences and the biology of agricultural crops, the genetics of agriculture crops to soil fertility and nano fertilizer. So a potential there as well for some collaborations.
Yes. So several research collaboration can be conducted in many land uses, forest, agroforestry, and also agriculture. Well, back to the presentation. We are now in the discussion session. Anyone who want to ask some question, please raise your hand by clicking raise hand icon in your Zoom room or type your question in a comment room. Is there any discussion? Or probably a committee have collect the question from YouTube? Because we are also live streamed in YouTube. Okay. Okay. There's a question from Anita. What is the relationship of tree composition? Tree composition on forest soil response to climate change, and what are the possible impact of climate change on forest soil health?
This is an interesting question. So I did not mention that we have a English oak dominated forest system that we are currently evaluating, but the co-dominants are sycamore trees and hazel. So we have a mixed system, so there are different tree species types. I didn't have the time to show the data for some of the sycamore and hazels in their responses, but our system is a mixed system dominated by oak. And oak is ectomycorrhizal, so its response to nutrient acquisitions would be different to the sycamore, which is arbuscular. And we do see that arbusculars have a more faster nutrient cycling responses than the ectomycorrhizal forest systems, which relies on the ectomycorrhizas to take up some of the organic nitrogen forms. So, of course, there would be different responses in terms of growth, but we are enlisting the NPP response in some of the nutrient responses of the other tree species that are within our fumigated rings. And if the tree species changes, there are changes in their responses. So absolutely there should be, but we would be able within our own studies to enlist at least father oak and sycamore and hazel trees in our systems. Forest health, it's an interesting one. In 2018, all of a sudden in the month of May, when the leaves came out, we had some moth infestation. So the leaves were constantly grazed by the moths, and we had a fall season in the month of May. So we were losing leaves early in the beginning. We don't know if what happened that particular year, but the infestation from the moths was substantial. And that's why we saw an increase in dissolved organic carbon in the forest litter. We currently have ash dieback diseases and other pathogens, and we have the oak canker disease. So our colleagues in biosciences are looking at the pathogens and diseases of trees and how they might be responding. So it's an interesting question. I cannot say how they might be responding, but there is a lot of work, and I mentioned that is a big project started recently called MEMRA. It's really to look at the memory and the response of these trees to diseases and pathogens, and whether they may have a memory to produce saplings which would be more resistant to fight the disease. So some cutting edge research is going on to enlist the response of forests to future the changes in diseases and infestations. And that's one of the key theme of the BIFAR research agenda as well.
Perhaps, Pak Cahyo, it can be our research agenda as well in UB Forest, right? Okay. another discussion from the student who followed this lecturing from Zoom? You can raise your hand to interact directly with Professor Samiullah. Okay. Another question from Amy. I'm interested in your study interest, how to join your research group as PhD student or postdoc fellowship. Okay.
So as
Professor
a PhD student, you could always apply to the department, and depending on your research interest, identify a potential supervisory team. Then the next question is funding PhDs. So the available funding that we have each year for PhD students is mostly for UK citizens. It used to be UK and EU, but after Brexit, it's mainly UK. Only 30% applicants from international destinations are still considered for those competitive PhD studentships, which are supposed to be announced in November. So look at the University of Birmingham website, PhD studentships in forestry and environmental science. With a strong portfolio, you could try, and 30% of the studentships will be allocated to international students. So there is that possibility to consider. And if you explore funding from your own countries or other international organizations, not sure if you have Commonwealth scholarships in Indonesia, the UK Commonwealth, no?
For PhD, I don't think so, but for master's study, I ever heard that they're kind of funding from Commonwealth country.
Yeah. Yeah. Postdoc fellowships are open competition, so whenever there is a position, people apply international and national. There is no priority. The priority is who qualifies the most and meets the essential criteria and bring in the desirable. So it's an open competition all the time, and there are always positions in different aspects. So jobs.ac.uk is your best portal to look for open positions in the UK academic sector.
Okay. Thank you, Professor. So, Annie, you can open the website of Birmingham University to find possibly possibly research funding from the university, they offered by the university, or simply apply to Indonesian government scholarship like LPDP, perhaps. Okay. Perhaps one last question. Is there anyone want to open discussion with Professor Samiullah? Oh, the question from Amy have been answered. None? Okay. Okay, well, ladies and gentlemen, we are in the end of the program because we have a very limited time also and no discussion again. So we should end the guest lecturing today. Hopefully we can see each other again very soon, online or perhaps offline after this COVID pandemic ended. I thank you very much to Professor Samiullah for your time and your great presentation, and also for all of the lecturer of Faculty of Agriculture and Soil Science Department, and all the students who follow this lecturing, both from Zoom and YouTube. I'm sure that we are achieving so many new knowledge and experience today. Before I close the program, I would like to request Professor Ullah's time for a while for joining with lecturer staff of Soil Science Department to discuss about potential collaboration in research and publication that we mentioned before. You have also research interest in forest, and we have a forestry study program, and we have forest facilities to do a research in the similar topics. Okay, and for all of the students, please leave the Zoom meeting. Finally, I'm Novalia Kusumarini, the moderator of today's program, Us for Leaving the Meeting. Wassalamualaikum warahmatullahi wabarakatuh. Good afternoon.